So just a word of warning, some of this talk is taken from what I gave up at Iowa State at their plant breeding symposium. I didn't want to give 100% copy of that, so I cut parts of it and I put some other parts in. And I know I have more slides than I should have for this talk, so I may not even get to the last part. I basically broke it into, first part is cotton, more important part. Second part, no, not really. Second part is sorghum, and I may or may not have time to get to that. If I don't, I apologize. It also is a very interesting story, and just so you know, I actually like all crops. I just happen to work a lot on cotton and sorghum, and there's a lot to be said for all of them. So I'm going to start off with just from a philosophical, this is where I come from. When I think about what we're doing, I'm really thinking about the position that society is in worldwide, really, not just the U.S., but certainly the U.S. is included in this. We have a big challenge to provision ourselves with all kinds of things that come from plants. And we've built some really tremendous agricultural production systems, but quite frankly, we're in a bit of trouble because in a number of locations, those production systems are really wreaking havoc on our environmental system, and I think we need to contend with that problem and do our best to modify those systems. That being said, we really need to provision for ourselves, and plant breeding is a big part of this recipe that we have for trying to do that. And it's not an answer in and of itself, it's not a panacea, but it is definitely a big part of that picture. When I was president for NAPB, a sustainability document put out by the chief scientist for the federal government on sustainability, they didn't even mention plant breeding. And I was just mortified, and I wrote a letter to her saying, this is ridiculous. And actually now they've gotten to the point where they're really thinking about plant breeding a lot. So take it to heart. You're working in the right area, in a good place today, plant breeding circle, and we have a big challenge ahead of us. So in order to address these things, we need to have genetic variation, and that's where I come from. That's what I'm thinking about. How do we capture genetic variation? How do we introgress and take advantage of naturally occurring genetic variation and work with it at a genetic level where we can actually extract the parts that we need and separate it from the parts that we don't need or which are actually deleterious. So just again, we have problems, we need to solve them. Another point is, I really think about plant breeding as a translational science. We take advantage of a lot of different things in what we do. And so it's incumbent on all of us to learn as much as we can about all those related sciences so that we can maximize our productivity. And, being in a major university, you really have an exceptional opportunity to do that. So I encourage you to fully indulge in this opportunity. So the needs for cotton are really not that much different from other crops other than we're dealing with a fiber. And it's not just that the fiber is not the only thing that's important, the seed is also important. But the fact that the fiber is an industrial commodity, it's traded in the international market, we not only compete with other countries, but even more importantly, we compete with synthetic fibers in the textile market. And that is a really, really big challenge because you're trying to pit a biological entity which is highly variable against things that are man-made, and those have certain advantages in that context, that way. So, if we're going to be using genetic variation, just to step back for a real quick moment, think first of all, when we're talking about elite genotypes that we use for agriculture, it's a very small part of what we would call the primary gene pool. And that's really an overrepresentation of the size. This is very, very small because we're dealing with very elite types that have been highly selected. When we talk about cotton and we talk about the primary gene pool, we're really talking about the tetraploid species, which would include what we allude to as the AD genomes, which are represented by this white area. They have 52 chromosomes, and I'll get more into the jokes in just a moment. But I wanted to mention that there are diploid species that have half as many chromosomes, and these have different genomes that are respectively designated by letters A through K without H being in there. So these genomes have geographically related distributions. The difficulty in using these germplasms is related to the genomic nomenclature. Just a word about that genomic nomenclature. It was really defined based on meiotic affinity, that is, during the reproductive cycle, the cells go through meiosis. And these different genomes have different degrees to which they will actually pair up and recombine with each other. And that is reflected in these designations or genomic groups. And again, they have a geographic relationship. Oops. So there are about 50 species overall. These numbers have changed a little bit because there's been one or two species. One's been described and one is in the process of being described, I believe. So we're probably up to 7 different 52 chromosome or AD genome species. And there's about 45 or so other species broken into these different groups. But again, the main point I wanted to mention is primarily talk about the AD genomes, the primary gene pool, and a little bit about the secondary gene pool. The primary gene pool is relatively easy to use. That is, you can make crosses pretty readily. There's some interspecific sterility and incompatibilities that come to play as you inbreed off of that. But by and large, they're pretty usable. The ones that come in from the secondary gene pool are more challenging to use. They require some special methods, and I'll have an example of that as part of my talk. And we pretty much, we have worked a little bit with this tertiary gene pool, but it's a bear, and we pretty much have been staying away from it since then because it's a relatively difficult and big challenge relative to the amount of work you put in there. You don't get a whole lot. So what about genetic diversity? I said you really need to have genetic diversity. And if we look at the cultivated cottons, this is an old piece of work that was done by Ed Lovers based on SSRs, but nonetheless, and there are lots of other studies that kind of show the same thing, with this one's suits my needs quite well. Shown in, these are all different genotypes shown on the right side. These are elite lines and cultivars and so on that have been SSR genotypes, so DNA markers, okay? And what it shows is that the distance of difference is shown in this direction, and you can see that these little black lines are pretty short. And what it's showing is that all of those are very similar to each other. And if I take a blow up at the top here, these are still the cultivars and so on, and then there's a palmari. Palmari is a race stock. It's a wild type that comes, it represents, if you would say, the non-domesticated types. And you can see it comes out a little bit further. But then you go up here and you see this is Pima and this Darwinii. So this is Gossypium barbadense, this is Gossypium darwinii. Those are other species of the primary gene pool. They also have 52 chromosomes, just like the Levi cotton that you and I wear. Barbadense is the $100 shirt cotton. Hirsutum is the $35 pair of pants and $50 pair of pants. Now, but you can see that the distance here is still pretty short, but it's more than it was for the elite types. What you have coming out here is when you go to these other species, you see this big jump out. Going from Hirsutum, you jump out and you see the other species come way out here. That's still the primary gene pool. If you go out in the secondary and tertiary gene pool, it goes way far outside the room, okay? So we're only tapping a little bit of overall genetic diversity when we stay within the primary, within the species and even within the primary gene pool, we're still pretty restricted. So, what do I want to do? I want to be able to transfer genetic diversity into the cultivated species Gossypium hirsutum, and I want to be able to use that genetic diversity. It's not as easily said as it, or easily done as it is said. There are a lot of roadblocks, if you will, that are put up by things that have arisen through evolutionary divergence, right? So how do we do that? Well, one route to that is to study the genomes, define them better, develop tools that allow us to use a more scientific kind of approach. People have been working on this stuff for a long time, and they haven't gotten a whole lot out of it. It's because of these roadblocks. So we have to find ways around these. So I kind of think about three challenges, if you will. So one is that traits of interest are very often very complex genetically, and we really don't have a good idea of how to approach these. Hung Bin does a great job of talking about this and really getting across the idea that, oftentimes you can't think in terms of one gene. We need to be thinking in terms of networks of genes and how do they interact and so on. Secondly, how do we combine genes that we're interested in? That can be a real challenge because they may be similar to each other, so it may be hard to differentiate their effects if you're trying to combine them. For example, if you have disease resistance genes for a given trait, you try to put them in the common background. It's hard to know if you have just one that's giving resistance or two, right? But if you have those tagged with markers, for example, then you can pyramid those genes relatively easily. Another problem is that genes are often cryptic, and so this is particularly important when you're doing wide cross introgression. Unfortunately, the way it is and has been for a long time at the federal grant level, you really can't put in a proposal and expect it to be funded if you just propose to introgress germplasm from a species A into a cultivated species B based on the assumption that there must be good genes in there and we're going to find them once we get them in. Rather, the only thing they'll really fund is if you already know that species A already has a valuable gene, such as a disease resistance gene, and you say, I want to introgress it, and then they'll say, okay. But the problem is, we've known for over 100 years that epistasis is a major player in complex traits. And that means that very often you don't know what the valuable genes are unless you have it in your background that you're going to use it, which means you need to move the germplasm into your cultivated background in order to evaluate what kind of genetic derivatives you might be able to pull from that. And then I mentioned, again, that because we're dealing with interspecific things, there are a lot of differences between these genomes that can create genetic hurdles that we need to be able to get around. So, cotton, as I said, has 52 chromosomes. And then I've illustrated another species, so this would be the Levi jeans cotton. This is the $100 shirt cotton, or Pima cotton, Egyptian cotton, whatever you want to call it. They have these designations. These are often written as subscripts, AD1 and AD2, to allude to the fact that they have this composite genome now that arose about 1 to 2 million years ago, actually from a common ancestor, as far as we can tell. And they did that by combining a D genome from something from the New World. We don't know exactly where that was. The closest extant species is held by many to be Gossypium ramondii. But anyways, an ancestor with a genome very much like the D genome that exists now in many of the Mexican species. And Ramondii was combined somehow, we have no idea, with a genome that came from Africa, or perhaps Asia. And like I say, 1 to 2 million years ago gave rise to a polyploid, probably through sexual polyploidization. And that evolved into various tetraploid derived species, of which we have somewhere between 5 and 7 recognized today, still living. In addition to those A and D genomes, if you think back to that chart that I listed with the different genomes, we have other genome designations that we find in various parts of the world at the 26 chromosome level. At the 26 chromosome level, there are actually 2 species that are also cultivated. They tend to be cultivated in rather challenging environments, fairly xerophytic. And these are Gossypium herbaceum and Gossypium arboreum. So this is what the genome of cotton looks like during meiosis, or the reproductive cell development, at a stage where the parental chromosomes have actually paired up. So there are actually 26 of these. They're called bivalents at this stage. And each one of these genomes, before it's replicated, consists of about 2.6 billion base pairs. Okay, so it gives you an idea. There's a lot of DNA there, and there are a lot of genes. This is what the chromosomes look like in a somatic cell if you catch them in the middle of the mitotic cycle at metaphase of mitosis. So there are 52 chromosomes, and so there's 26A, 26D chromosomes. And this same kind of picture would be seen if you did any of these other AD species. It'd be very, very similar. Not identical, but very similar. So this is like a deck of cards, right? That's what the easiest way to remember it. So cotton is like a deck of cards. We can all go gambling and try to figure out how to play the genes right. So the way it is right now, we allude to the A genome chromosomes as numbers 1 to 13 and the D genome 14 to 26. There's not a exact one-to-one correspondence between these because of some translocations, but by and large, you can find either one or two chromosomes that corresponds to a given chromosome from the other genome by way of evolutionary origin. So it's kind of interesting if you take certain repetitive sequences and you do fluorescence in situ hybridization. So we actually label up that DNA so that we can then localize a fluorochrome where that DNA hybridizes to the chromosome. So we take a sequence that is repeated within the genome and hybridize it. We can look at it in the diploids and the tetraploids, and a few of them we'll find behave like this. And so this really nicely depicts this tetraploid architecture. And I don't know if you can hit the light back there for just a moment. Wake up. So here's an arboreum, a diploid species with 26 chromosomes. Here's Gossypium ramondi. Here's another D genome species. And basically, you don't see it present in the D5. You see it's very heavily present in the A genome. If you look at the tetraploid, so this is Gossypium hirsutum, you can see half of the chromosomes are lit up with it and half are not. So what does that mean? That means that this particular repetitive sequence was present in the ancestor at the time when this tetraploid formed, and it has not moved from one genome to the other. It's been immobile, so it's like a tattoo on the genome. And this is actually the rarity. Most of them have moved around. This is quite interesting. But nonetheless, it shows you this architecture very nicely. Could you turn the light back on, please? Back one. Okay. Whoops. So that kind of fits this idea that we have the deck of cards, but what I want to get across is that cotton is really probably more like a pinochle deck or something, or even worse, okay? Because we have higher levels of polyploidy, and this is something that we really got into in the 1990s, and it's gotten to be a real fashionable thing again here the last few years, talking about this, but we became quite convinced of this quite a while ago, and I'd like to show you just a couple lines of evidence that I'll point out. So one is the base number in the Malvaceae, the family in which cotton belongs. It has a base number around 6 or 7. Secondly, we did some experiments with bacterial artificial chromosomes, which are big pieces of the DNA out of the chromosome that have been cloned, and we can hybridize them just like those pictures I just showed you and see exactly where it came from. And we could see that there's some redundancy there that would only be occurring if cotton has actually gone through more than one round of this polyploidization. And I mentioned that I'm not going to show you slides of this, but we actually have some fish slides that if you look at them with certain repetitive sequences, you can see even an older pattern, much like I showed you, not quite so demarcated, where you have a differential between half of the genome of cotton being strongly labeled and half of it not, for subgenome. So half of the A genome and half of it not. And then third, I'll show you a diagram that came out of one of the papers with Andy Patterson when he was still here, depicting this also. So here's a picture of Sida. So this is another member of the same family. And this is from a paper, some work we did in the 90s with Paul Frixell, who was a taxonomist that used to be here with the USDA. And it just illustrates very nicely a case where you have 7 bivalents. which means the base number is 7 for this particular species. So many of the species you find 6 or 7 in the Malvaceae, not 13 like it is in Gossypium. So here's a picture of Gossypium arboreum. So this is a putative diploid, okay? People hold this by dogma to be a diploid, but it's not, right? It's a paleopolyploid also. And so this also was done in the 90s or maybe early 2000s, but I think it was the 90s. And so if this is a diploid and I hybridize an RFLP probe, which is a single locus, single copy sequence, I should only see 2 chromosomes that have this particular sequence. This particular sequence is also synthetic, that is on the same chromosome as a nucleolar organizer, which is a nice morphological feature that we can see cytologically. And when we fish this, and maybe you could hit the light again, you can actually see there's small arms here, and then you see these pink signals. And so there's 4 of them. There's just 2 cells representing this particular species. So this, too, was another indication. It's synthetic also in both cases with the nucleolar organizer, and it's occurring twice as many spots as would be expected as in a diploid, indicating that this is, in fact, a paleotetraploid. Lights, please. Third line of evidence came, like I said, from work done with Andy Patterson's group as part of the RFLP mapping project, which is really the first real high-quality genome mapping work done at the molecular level in cotton. And one of the charts in the publication Reinisch et al. Is this one. And it shows the number of fragments that you get after EcoR1 digest, which if it's in these cases you would expect maybe to get 2 different fragments out of a homozygous line, one for the A genome, one for the D genome. But in fact, the average number is 4, again indicating a higher level of polyploidy. So there's really no question quite a while ago that cotton was a higher level of polyploidy or paleopolyploidy than was customarily thought of. And I think this is, people have come to realize that this is generally true of plants today. And in fact, the story gets even more complex. Patterson stayed on this and recently put out this new paper in Nature where he talks about this, and he alludes to the evolutionary origin of cotton, showing very, very old paleo 6x ploidy, so a 6x doubling or increase, then a 5 to 6-fold increase, and then some more coming up here in Gossypium. So you can see this is something that's occurring again and again in plant genome evolution. And so we're just kind of looking at the end of the process. We're not looking, and unless you can get in there with molecular genetic sequencing, you can't see all this detail, but it's there. So this model is inadequate as well. But it underscores why plant genomes are so complex, okay? There's a lot of redundancy in there. A lot of it gets sifted out, jettisoned, but there's still quite a bit in there. So one of the things that happens as a consequence of this ploidy, and so things can become more diploidized or they can stay at a higher level of ploidy. When things are still at a higher level of ploidy, they become relatively tolerant of variations in chromosome number, where that chromosome variation in chromosome number affects genetic balance. Okay. This doesn't happen a lot in animals because of the sex determining system, but in most cases in plants we don't have to deal with that. And so plants this is pretty rampant in plant genome behavior. And what I just made this little diagram to kind of indicate that as things are more diploidized, they get less tolerant. And secondly, that there's somewhat of a gradient here between something being intolerant. The plant may be viable, but it may not be able to transmit a deficiency or an extra chromosome through a male or a female gamete. And there's a big difference between the male and the female gamete because male gametes actually express many more gene in plants. I shouldn't say gametes, gametophytes, right? The pollen expresses many more genes than a typical megagametophyte, right? And because of that, it's more sensitive. And so what we find is that we can actually transmit these aberrant chromosome numbers more easily through the female gametophyte, the megagametophyte, than we can through pollen, and that's generally true. And then there are cases where they pretty much tolerate everything, and sugarcane kind of falls in that category. So, just kind of summarize what I've said. Polyploidy and paleopolyploid are prominent features of cotton, right? There's a number of lines of evidence for that. And some of the ramifications of this are important. So it complicates inheritance, complicates anything having to do with molecular genetics, including genome sequencing and assembly. It can complicate and reduce the rate of genetic gain based on phenotypic selection. It can complicate the development and use of markers, of which I'm all too familiar because we've been working on that for a number of years. It, however, facilitates chromosomal manipulations, and I'm going to talk about those a little bit because few people in here take cytogenetics, so I'm going to force you to have a little in here. And a big thing is that it enables female transmission of conditions where they're actually missing a chromosome, so monosomics and segmental deficiencies, and we've taken advantage of that in our work. So just a kind of a outline here of some things that we've been doing. I'm going to dig into detail in a second here. So we've developed conventional cytogenetic stocks of Gossypium hirsutum, things that are missing specific chromosomes and segments. And we've used those and are using them for a number of different things. We've used them for mapping of genes. We've used them to breed other hirsutum aneuploids. We use them to create interspecific aneuploids. I'm going to talk more about this in a moment because we've used those for large-scale mapping and binning, and we've used those for chromosome substitution. Very recently, we've developed some technology that allow us to do ad hoc copy number variation analysis, and so I think that this is an area that we're probably also going to apply these. Oops. Hyperactive thumb here. So, I've mentioned hypoaneuploids. This is a definition slide, if you will. This indicates a normal cotton would be called a euploid. That's a true ploidy, right? U meaning true. So it has 52 chromosomes. At meiosis, it forms 26 bivalents. So it would have 26 of these things. This picture is actually of a monosomic that is missing one chromosome. We don't name it for the missing chromosome. We name it because there's a single chromosome left there. But we call this a monosomic. And so rather than having 26 bivalents, it has 25 paired chromosomes or bivalents and one univalent. So a total of 51 chromosomes. Then we have some others that are missing parts of chromosomes and a little bit more complicated types. But I'm going to skip over that because it's not germane really to what I want to talk about. I do want to point out that these different aneuploids or hypoaneuploids have syndromes of morphological changes, much like a Down syndrome human has a syndrome. You can always spot a person that has Down syndrome very easily. And you should know, by the way, that those are very select people, so be nice to them. Only about 11% of Down syndrome kids or conceptuses survive. So they're the top 10%, right? Keep that in mind. So anyway, these things have very distinctive morphology. It's still a challenge to see them in the field where there are lots of environmental things that can affect the phenotype. And their transmission in cotton can range from close to 0 to close to 50%, depending on which one you're talking about. So some are easy to work with and some are really difficult. So we've developed a number of these for whole chromosomes, which are depicted here, as well as these are called telosomes or monotelo-disomics for different chromosome arms, either the short arm or long arm. And we've used these then for various kinds of things. In order to do that, once we found them, we back-crossed them to a common genetic line, TM1, which is a genetic and cytogenetic standard, so that we'd have an isogenic platform from which to work. And that's what has been quite helpful. One of the things that we've done with these is use them for the as recurrent parents and modified backcross programs to create chromosome substitution lines, or what we call CS lines. And this is done pretty simply. So this is depicting a monosomic that's missing one copy. It only has one copy of chromosome 4 instead of 2. And that one always comes from the male through the pollen when we recover it. We use that as a female parent, and we cross it with a donor genome. And then we recurrently use that same monosomic type as the female parent during the backcrossing program. And at each generation, we purposely go in and we find and select a monosomic individual. And the reason we do that is if we have a monosomic individual, there is no pairing partner with which to recombine for that one chromosome, whereas all the other 25 chromosomes can recombine, and they will. So each generation you're having recombination throughout the rest of the genome, but not this one monosomic chromosome. So it's maintained in an intact state during the backcross process. Meanwhile, you're getting rid of the alien genome as you backcross, just through segregation as well as recombination. So down at the bottom would be a idealized version where basically we have only the alien chromosome. We get down the bottom, we inbreed it, and we select for one that has 2 copies, and then we essentially have one alien pair of chromosomes and 25 of the recurrent hirsutum parent. So this would be a chromosome substitution line. So just depicting it again here in this case for chromosome 25. Each one of these has a composition that is therefore quite predictable, and that's barring human as well as biological errors, and both can occur and do occur. And so a little bit more about that in a moment. But if they're good, we should have somewhere between 500 and 4,000 genes of alien genes in there in each line. They should be pretty close to quasi-isogenic or isogenic with TM1, the recurrent parent, as well as with other chromosome substitution lines, right? They're true breeding, so I can increase the seed, I can run replicated experiments, and for some kinds of experiments they're quite nice because they reduce the genetic noise, and if you think about it, you remove all the interactions with the other alien donor genomes for 25 of the 26 chromosomes, and so it kind of simplifies the analysis. So we've used this approach, and we've developed chromosome substitution lines for Gossypium barbadense, Gossypium tomentosum, Gossypium mustelinum. So this is your $100 shirt cotton. This is a $0 cotton. It's a wild Hawaiian species, good for nothing, and it looks nice, and it's kind of a cool plant. Very grayish looking because it has a very pilose leaf, really dense surface trichomes on it. And it's been used for some kinds of breeding in the past. And then Gossypium mustelinum, which is a Brazilian wild species, also non-cultivated. Nonetheless, each of these has genome architecture very much like cotton. So it's not a comprehensive coverage, but nonetheless, still valuable, okay? So these collectively constitute our chromosome substitution platform. It's isogenic. It thus provides introgression materials that can be used for breeding as well as for genetic analysis. Oops. So what can we do with these things? Well, we can analyze the lines per se and get an idea of what kinds of phenotypic variation and how much of that we can ascribe by analyzing a bunch of these at once. We can ascribe which parts of that variation are due to genetic causes. We can get a little bit more detailed if we work with more advanced generations, F2s, F3s, F4s, and so on. So we get an idea of the additive genetic variance, which is more important from a breeding standpoint because that's the stuff that's easier to account for in a breeding program. If we go into multiple generations, we can get at what I think is probably the most, in some ways, the most important, at least from a wide cross breeding standpoint, epistatic effects. And the reason I say that if you're working with interspecific things and you're dealing with wide cross integration, there are an awful lot of genetic interactions that come into play. And so the problem is that you often find that what you expect is not what you get, okay? And that is because of epistasis. And that's another reason why I think having support to introgress material is important, because you need to do that in order to see how valuable it is. Anyway, so if we go to multiple generations, we can get after epistasis. If we use top crosses, we can get after the epistasis as well. You can look for cultivar to chromosome substitution line interactions. We actually got a new project started up with Wayne Smith's lab with Damon, and he's going to be working this a little bit. Another one is chromosome substitution by chromosome substitution intercrosses. Again, getting at specific chromosome-chromosome epistatic interactions, which is kind of interesting, because if you contrast that with the normal approach, which is look at the whole genome at once, you have an awful lot of noise going on there. And so, in a way, this is like coming at the same problem in a different, more simplified, reduced data set. And so it's going to be interesting to see how this plays out. The last one I want to mention, and I'll actually illustrate this in a minute, is to go after chromosome-specific RILs, or recombinant inbred lines. I'll get to that in just a moment. But I just put in one chart here just to illustrate a few things. So this is using F2s, and it actually shown here is the type of genetic contribution. This is for seed cotton weight. This is lint, so this would be lint plus seed. This is lint alone. This is lint percentage, so the amount of fiber, if you will, over the seed cotton weight, that is the seed plus the fiber. This is the boll weight, micronaires, and various traits. And all I wanted to point is that for different traits, you're going to see different patterns of inheritance, right? And some of them show relatively large amounts of additive variance. Others are more dominant variance. Others show more interactions and so on, which is not surprising. One of the neat things about these substitution lines is once you have them, you can send them to any collaborator who's willing to analyze them. And so I'm going to illustrate with a couple of charts here, a recent publication that goes beyond just the fiber traits and agronomic traits that were just talked about. This goes after specific things, and this is for resistance to FOV, or Fusarium oxysporum basinfectum, which is a pretty big problem in some parts of the world. It's gotten to be a pretty noticeable problem in California recently, because they have a new race out there called Race 4 that's wreaking havoc. So this chart actually illustrates data for both FOV1 and FOV4. So FOV1 is the more common one that we've had in the U.S. for a long time. This one doesn't cause much of a problem unless you have root knot nematodes through which it gains entry into the root and then into the vascular system, where it wreaks some havoc on the plants. This new one, however, doesn't require the root knot nematode. It just digests the roots, right? It attacks from the outside, and so it's really, really nasty. And so we sent to Mauricio Oyoyo, who's actually used to be in California, but now he's up in Lubbock, but he's still conducting the research out in California with another collaborator out of there, Phil Roberts. And he evaluated a bunch of these chromosome substitution lines out here, and the gray ones represent FOV1, and the black ones represent FOV4 susceptibility. So this is more susceptible at the top, less at the bottom. And it's not too important to see the exact numbers or anything. It's just that you can see there's variation here, okay? And the blue ones actually have the lowest levels of FOV in the field, and so it just shows that there's significant genetic potential in these materials. Likewise, you can find some that are terrible. This is chromosome 17, and both these are greenhouse data. These represent field data actually down here. So these were low in both the greenhouse and the field. This was high in both the greenhouse and the field. And it's just to illustrate what can be done. This is with root knot nematode. And so it's pretty much the same set of lines, but same idea. You see variation. Here's a couple that are both better than donor. These two are better, or same, different assay. This is for galling data. This is for egg production. And actually the same lines were better in both cases, in this case relative to the donor, here relative to other chromosome substitution lines. So you can take these lines and send them, look for temperature sensitivity, like for pollen fertility or drought resistance, whatever. And so that's the kind of mode we're in now, is trying to develop collaborations with people to look at these various kinds of things. Up to this point, we've relied pretty much on quantitative statistical genetics, but now, as I'm going to mention, we've developed some nice SNP technologies, and so we're going to be morphing over into more sophisticated types of analysis with that. And in that light, I'd like to mention these chromosome-specific RILs that I've mentioned. So this depicts a couple of lines, normal RIL lines, which would arise after a number of generations of selfing of an F1 between two parents. So this would be the first generation of selfing. It's going to be about 50% of each. It's going to be homozygous and heterozygous for various segments. But after you inbreed, everything is homozygous for something, or one parent or the other, for various segments. And so this line is homozygous for a bunch of stuff. This one's homozygous for a bunch of stuff, and it's just kind of random as to which ones are similar and where they're different. So normally, when one's going to analyze these, you have 100 or 200 of these. 200 would probably be a better number to analyze in order to try to figure out what's affecting what trait. Well, another approach, which is not better than this, it's complementary, I think, is chromosome-specific RILs. And so the idea here is that in an idealized fashion, we have a disomic chromosome substitution line. I missed the chromosome here. So there should be another white one here. So it should be heterozygous here. And then when you self it, you end up with segregation for various segments here, but there should be homozygous in each of these lines. And so with 30 sacs is probably enough, but 30 to 50 of these things, you can get a very good idea of what's going on in terms of the genetic contributions to this chromosome, because you don't have all the noise, if you will, that comes from all the segregation around here. So it really makes for a very, very nice analytical platform. We're extending this technology, including the use of SNPs. So I asked a couple of my students are working on this. Yu Ming, Lynn, and Luis Santiago are working on this project where we're starting to apply some of the SNPs, and so I asked them to send me it's an early draft of a map that's been made off of this chromosome 17 based on a set of 49 RILs, which actually they've already been phenotyped, and so we're just trying to come back at them with SNP analysis. And these were created and with CASP that we developed over here at the AGL, Agrogenomics Lab. So if you're not familiar with that, you need to check into that facility. And then what we did was we took those and we went over to the TIGS facility, which is on campus, and they have a fluidyme system. We use that to run the markers because it's cheaper for if you're doing a lot, like we were here. And then created actually we're using 3 different programs for mapping, both join map, IC, and then R, because we want to see how they could stack up relative to each other. So anyway, I thank those guys for sending that over to me. But it illustrates the fusion of technologies, the molecular genetic marker technology with the advanced chromosome substitution platform. How am I doing? I'm getting near the end. I'm not going to make it to the sorghum. So this is just to kind of summarize where I've been. We've identified here pseudo aneuploids. We created these isogenic stocks. We used 3 different donors. We created these chromosome substitution lines. We've been proofing those, if you will, validating them with a couple different kinds of marker technologies. Now we're applying SNPs. Basically, this provides us about 100,000 alien genes at this point in this isogenic platform. We also use these and released 17 of the barbadency lines. We haven't released the other ones, but we have conducted a number of complementary kinds of statistical genetic analyses to look at genetic control over various traits. For this platform, there are several things that we kind of expect to do. One is to continue our analysis and really detail the validity of these things, and also to characterize the genetic background, because we know that there's probably pieces of the donor genome still stuck in the background. We'd like to know where those are and when they're segregating, if they might be influencing the traits that we're interested in. And we might discover some other things, like it's very likely that there are some that are present in many of the substitution lines because they flank a gene that is preferentially transmitted through one sex or the other, and so during the back-crossing process they may be preferentially retained or lost, as the case may be. So we're using SNPs to enhance the analyses of these, and we're extending this into these different other areas, and also the collaborations used to examine different kinds of traits. In addition to the chromosome substitution per se, we've also developed a parallel project based on marker-assisted selection where you can use linked markers to introgress segments. So rather than doing whole chromosomes, we're now going after multiple segments out of a given chromosome using several markers for each segment to back-cross that in. This is an approach that was developed actually in tomato from some Israeli individuals. And it provides an alternative method of introgressing, and so what we're doing is we're looking at that as a way to complement the chromosome substitutions, because we don't have complete coverage. And so we're going to go after the segments that are not covered by the chromosome substitutions. In addition to that, we've also decided to use that technique. So Amani Kainam, who's here, raise your hand, is working on this project. So we're doing the same thing with a synthetic tetraploid. So to bring in some secondary gene pool germplasm, okay? And to do that, we're using some, I'm going to jump through this. Whoops. So I just wanted to mention, so about a year and a half ago, we released the cotton SNP chip, which is the culmination of about 5 or 6 years worth of work. And so the original array had was synthesized with about 70,000, and then it's typical, about 90% of those worked and about 10% didn't. So the array has about 63,000 active assays on it. Of these, about half, or a little bit over half actually, are working pretty well. And they provide a source of both intraspecific and interspecific SNPs. So this chip, so each one of these assays includes, you can run 24 of them. This is a microscope slide, by the way. So that microscope slide costs about $2,500, not quite, about $2,000. But you can run 24 different samples on that slide, and each one is going to be analyzed by these same sets of molecular genetic DNA markers. So this slide is just depicting that once we developed this system, we had to create a statistical analysis of all of these, and this is done in conjunction with a European company, Trait Genetics. They created what's called the cluster file, and this is good for doing analyses of parental combinations involving hirsutems only, or hirsutems with other AD species, such as Gossypium barbadense, Mustelinum, and tomentosum. It's not designed to work with anything involving the diploids. And I want to mention that because Amani's actually done this for us since then to create one specifically for diploids, because she's working with diploids and this one doesn't work for her. Anyway, those are important, because once you have these, you can do automated genotyping. You just stick your data in there and the computer spits back the genotypes. So in the case of the initial work, we actually created a couple of maps for hirsutum. One is intraspecific, one is interspecific. This one's close to 20,000 markers on this map. This one has about 8,000, and that's pretty typical for any parental combination of hirsutums. these are very, very useful for all kinds of purposes, and people are really starting to run with this technology now and use it for all kinds of things, and we're amongst those groups. But there are many others that are doing this also. So I only have a few minutes. I'm going to breeze through one last topic because it's very different in kind of composition relative to the chromosome substitution. It illustrates another kind of effort related to wide cross introgression and utilization of naturally occurring germplasm for breeding purposes. It also illustrates some of the kinds of difficulties you can run into. So we got involved in this project as a collaborative effort with the USDA group here, with the plant pathology group and nematology group. Forrest Robinson was here at the time. Al Bell were my primary collaborators on this, and then I had a student that came in, Neelish Dighe, that became closely involved with this project, and it was geared towards developing resistance to reniform nematodes, which have gotten to be a pretty big problem in the U.S., particularly in the southeast, but definitely also in parts of Texas. So these little critters are soil-borne nematodes, and they latch on, the females latch onto the roots, and they can do quite a bit of damage, as illustrated here. And so this illustrates some of the kinds of stuff that you can do with cytogenetics. A synthetic tetraploid was actually created, which has 3 species in it. And I think because time is limited, I'm not going to go into the detail, but it has, one of the genomes here is from an African ancestor, which was known to have extremely high, the highest resistance to this reniform nematode, and that's why this tri-species hybrid was used. So this is used as a parent then, donor parent, in a modified backcrossing program. And I say it's modified because we infused the use of cytogenetics in it, and then as we got things better defined, we infused marker-assisted selection into the process. So the actual program changed across as we got more information and went along. Ultimately, we extracted a bunch of inbreds, and a couple of those were actually formally released, and even before they were released, we sent them out to people so that they could start growing them and see how they did and breeding with them and so on, beyond the environment that we had locally. This is the breeding pedigree that came through, so you can memorize that in about 3 minutes, right? So it is a mess, okay? But what we did in this project, we didn't set up specific families to do the genetics. We used material that was available and being produced to do our genetic work. And that's, you know, for somebody who's doing breeding, that's a lot better than having to create an ad hoc population. And so we coupled that with cytogenetics, and we showed that as we backcrossed, we could get back, by the time we got to the BC3, we were getting back to the 52 chromosome level. However, when we look at that, there are actually these both have the same number of chromosomes, but they behave quite differently at meiosis. So there are 2 modal types amongst these 52 chromosome types, one which had 26 bivalents and some that had 25 bivalents plus 2 univalents. And so basically this would be a hirsutum chromosome, and this would be the alien F genome chromosome. And they were all resistant, by the way. These were. So that basically comes about because these 2 chromosomes aren't pairing with each other, but this one apparently we've got the resistant segment in there, but it's homologous, and so it recombines with each other, and thus it forms a bivalent over here in meiosis rather than 2 univalents. This one can't form a bivalent, so they are present as univalents. And if you don't understand this, you better take cytogenetics. We also were able to determine by looking at these, and you can do this cytologically, that there's something on the order of about 10% recombination, 10% of their normal rate of recombination. So we knew it was, the important thing is we knew it was markedly depressed. And that's very, very common when you're doing wide cross introgression. And that means that getting recombinants where you want is more difficult. Okay. We did the mapping. At this time, we're still working with SSRs, and so you can see some associations of susceptible resistance and difference in the patterns across here. So we had marker associations, we created linkage maps, and then we it was kind of fun because, you know, we were doing our marker stuff, and we were trying to tell the pathologists, you know, you really should do marker-assisted selection. They said, no, we want to do the phenotypic selection. But then after a generation of comparing their results and our results, they switched over and they just did use markers. Because it's very difficult to do really, really good phenotype, particularly to detect the resistant, accurately detect resistant types is difficult. So we released these things, and you might think this diagram is mislabeled, but it's not. So we sent these off to our collaborators, and they said, call back, you got a problem. So if you put these things in a hot field with lots of nematodes, the susceptible ones do pretty, I mean, they're effective, but the resistant ones just really suffer. And so this is the resistant line, which is exactly the opposite of what we expect. Okay. So we took on a project to work on this, and we figured, well, we had several hypotheses that could potentially explain this, one of which was that you had a linked susceptibility locus that had been introgressed. So you'd lost a resistance to a soil-borne pathogen, a fungal pathogen or something like that, that was tended to co-occur with these things. And so we went about the genetics. We developed a bunch of SNPs. I'm not going to go into detail. We characterized these enough to where we had enough to work with using the CAS genotyping over at the AGL that I mentioned before. Whoops. And to extend our work on this, we really wanted to do large number selections because we wanted to have if the resistance gene was here, we wanted to have recombinants on both sides, right? That's not easy, particularly if it's hard enough in normal situations, but when the recombination depressed, it's even harder. And so we knew we needed large numbers to do this. And so we kind of, all right, well, what can we do? So we developed a non-destructive seed extraction protocol that, one, is important, it's non-destructive. Secondly, it's pretty high throughput. Third, maybe most important, it's cheap. Okay. So a normal DNA extraction will cost you anywhere from $150 to $350 if you buy QIAGEN kits or something like that, or macronatal kits. So this costs 5 cents. And so this student, Shuting Zheng, who was a student at the time, did this, helped perfect the technique and then applied it. And in 3 weeks, she made 20,000 DNA extractions and the genotyping, and she found 5 recombinants from this. And so it's a really, really powerful approach, okay? It's even faster, by the way, if you go to seedlings, but you have a time constraint once you have seedlings. And so she had 5 recombinants out of 20,000. So it gives you some idea of the selective power, right? Unfortunately, none of them had got rid of the stunting. So we didn't make any progress other than the fact that we established a really, really valuable technology, right? So, all right. So I'm going to wrap up. Many thanks to, and this is just the most recent couple of renditions of our lab. I'll also cite a few people down at the bottom that have contributed to various parts of this. But many thanks to the people that work in my group. I mean, they're the ones who do it. I just push papers around. Lots of important collaborators. Just kind of categorically specific people working with the chromosome substitution lines. I'd like to mention Sukumar Shah, who was actually one of my first students, and his research director, Johnny Jenkins, have been really, really big in this. Mauricio Oyoyo recently, and for the pathology, Phil Roberts out in California have been really big for the chromosome substitution analysis with the FOV and root knot nematode. A lot of the SNP work was done in conjunction with Alan Van Dynes and Hamid Ashrafi from UC Davis. Sequencing, Jeff Chin is leader of our NSF project, and so that had a lot to do with some of the SNP work as well. For the reniform, I'd like to particularly mention again Al Bell. I don't know if I don't have, I should have Forrest Robinson on here also, and then people that he works with as well. Shuting and Neelish were really big in this project, and lots of other contributions from our laboratory as well. And then lots of people for the SNP chip. And I should mention the SNP chip was not just us. We were a lead on it, but this is a worldwide global effort. So there are lots of people that were really important to this. But I'd like to especially mention Amanda Holst-Kemp, who many of you know. She was a student here and then a postdoc, and she's actually a postdoc with Alan Van Dyne now, but works in North Carolina. Alan Van Dyne and Hamid. David Fong contributed quite a bit. Mauricio donated some of the mapping populations. The CSRO group was extremely big in this because they provided the RNA-seq data for Hirsutum markers. We provided genomic SNPs for Hirsutum, so that was a really, really big contribution. And so they deserve a lot of credit for that. And Illumina was great in terms of the assistance that they gave us, and of course support from Cotton Corporate is important in all of these things. So, time's up. I can't go on to sorghum. Sorry, sorghum. Yeah.
We're actually short on time. So if people have classes to go to, you can go ahead and leave.
Yep.
I'm sure he's willing to stay after though, and he'll take individual questions. And then also it also gives a great opportunity because if you have more information on sorghum, we can bring you back. It's that way the sorghum folks get some attention as well.
Good.
So.
David, a lot of work.
A lot of good people. You were one of them.
Man, I do not know how you achieve that much. Yesterday I was reading an article. So one group of living organisms, they don't sleep. The human do sleep. I don't know whether you have genes of this category or not. Anyway, you said that the substitution line is the finest way to do the breeding. I do believe
No, I didn't say that.
Well, I do believe that. The mass is not there, just only 2 chromosomes.
I think it's a complementary approach.
It's a lot of work to do the substitution. It's a lot.
Yeah, but
to
with
create.
SNPs it's going to be easier.
Well, and also the other mass to clean it up.
Yeah.
Another questions I have, that's how many characters or characters been transferred from the Hawaiian tetraploid and Brazilian wild relatives?
How many characters have
We have in cultivated cotton.
Ooh. There's disease resistance genes, there's morphological traits, but just
So there's
I don't know.
some
there's
good
some.
contribution. Good.
yeah, I mean simply inherited things. Wayne may want to comment on this, but I wouldn't say that. Well, part of the problem is when you talk about complex traits, it's very difficult unless you have molecular data to actually detail what's been done. And so I think we'll know more. I'll be able to answer that better in another 5 years as we get more information, more sequencing data and stuff like that. But in terms of qualitative traits, I mean, yeah, there's some that have been done, but those are the only ones that are easy to say that, okay, that came from. An example would be the pilose trait that I mentioned for tomentosum has been introgressed. Joshua Lee at North Carolina did a lot of work on that. So.
And polyploid you mentioned, then Malvaceae has basic number 6 and 7. Do you think some long ago 2 species with 6 and 7 hybridized and got the 13 chromosome?
Exactly.
Okay.
That was my point. Yeah. Yeah. So the polyploidization, and usually it's sexual polyploidization, is a recurring phenomenon. Most of them don't make it evolutionarily, but a few do, right? And there are some predictive features of those sexual polyploids when they arise that would make it more or less likely that they would survive. One of them is that those genomes that come together need to be more or less meiotically independent of each other. Because and you know this because you're a cytogeneticist, but if they interact and are exchanging pieces and they are basically have different computer programs, if you will. If you're switching out cards from one program or parts of one computer program with another one, you're going to create a mess, right? what works is when those two sets of chromosomes, those two computer programs can run side by side and run the cell at the same time. But if you're having genetic exchange between them, if they're interacting meiotically and switching pieces, you're gonna have a mess, and it's gonna die out. Okay.
David?
Yeah. Anybody wants to stick around, you can stick around, ask more questions. But those of you who need to leave, please feel free. Because I know people have classes and all sorts of stuff to do.
I didn't have a question.
Oh, that's fine. Let people just exit and then you can have a question.